My name is Ayumu Nishimura, and I'm from Saint Maur International School. Before I start my presentation, who in here hates going to the dentist or don't like? Please raise your hands. Yes, some. Well, I don't either. So in my research, I investigated the antimicrobial effect of tea and its potential benefits on the oral hygiene, which may save us a trip to the dentist. Over 200 bacterial species exist in our mouth, and while some may be harmless, others are not. Indeed, species of Streptococcus mutans is one of them that have problematic effect on human health. Overgrowth of Streptococcus mutans can cause dental decay and gum diseases, having symptoms of bad breath, foul taste, and even tooth loss. And not only will the effect remain in the mouth, it can also spread to other parts of the body through soft tissues, leading to blood clotting or even mouth floor infections that may also lead to life-threatening complications, and that's scary. So I wondered if there's anything we can do in our daily lives to prevent such health risks, and found some literature suggesting that tea drinkers seem to have decreased risks of dental decay and tooth loss. So in order to further investigate this correlation between tea and dental decay, I decided to test the direct effect of tea on bacterial growth in the mouth. And while I hope to use Streptococcus mutans in my research, due to IB regulations not allowing manipulation of organisms from body fluid, I decided to use a model organism called Lactococcus lactis subspecies cremoris, which I'll refer to as L. cremoris from now on. L. cremoris is a safe organism often found in food products, and it bears over 90% genetic similarity to Streptococcus mutans in its 16S rRNA sequence. And due to its genetic similarity, it also has physical and chemical similarities in the fact that it's gram-positive, facultative anaerobe, and the morphology being coccus. And because of the specific morphology, I was able to isolate the specific pure culture of L. cremoris from the mixed strains of bacteria that I obtained from yogurt kit powder. And here we see a picture of both Streptococcus mutans and L. cremoris having chain structures. Now let's move on to the details of my study. Right here, we see a typical example of a plate that I used in my research. And I used disc diffusion assay during my experiment, which requires placement of filter paper discs that has been soaked in antimicrobial agents, in my case, which was tea, and other antibiotics. And this plate here shows a growth of L. cremoris on the plate after 72 hours of incubation. And as expected, we see that distilled water negative control showed no growth inhibition of L. cremoris, while the positive control streptomycin, which is antibiotics, had a clear large inhibition. And other discs soaked in teas also exhibited growth inhibition of L. cremoris, while they differed in sizes. So in order to quantify the inhibitory effect of each disc, I looked at the microscopic close-ups. And during this process, I found something very interesting. While the antibiotic disc showed no visible colonies within the zone of inhibition, discs soaked in teas actually showed visible colonies around within the growth zone of inhibition. And the sizes of the colonies also decreased consistently for the colonies around the disc. And so it suggested that while antibiotic can be killing the bacteria, having bactericidal effect, teas may only slow down the growth of L. cremoris, having bacteriostatic effect. So after I quantified all the growth inhibition by all samples, and here we see average growth inhibition by different types of teas. And we see that all teas did show some kind of inhibitory effect on the growth of L. cremoris. However, while there were large variability within samples shown by standard error, after conducting a statistical analysis, I found that jasmine tea, black tea, and green tea were not significantly different from each other, and all three of those together had significantly higher inhibitory effect compared to water, rooibos tea, and oolong tea. So I wondered if there's any compounds in teas that actually triggered the inhibitory effect. And first I looked at caffeine, and although caffeine did have a positive correlation with the inhibitory effect, it was a very weak one. And so I looked at polyphenol. And polyphenol did show a moderately positive correlation, suggesting that it could be one of the more important causes of growth inhibition. So in conclusion, most teas did inhibit the growth of L-coumarins, and the mode of action of inhibitory effect being bacteriostatic, which slows down the growth of L-coumarins. And in addition to that, I found that concentration of polyphenol in teas seemed to be moderately correlated with the inhibitory effect. So regarding the results of my study, as well as the previous studies, it seems that regular consumption of tea may reduce the oral density in the mouth, as well as save us the trip to the dentist. If I had more time and funding, I would like to use actual species of Streptococcus mutans and also focus on direct effect of polyphenol on the growth inhibition of L-coumarins using different types of polyphenols and different concentrations. And also I would like to optimize the quantification method by using fermenters in liquid culture, as I found that there were variabilities in tea diffusion on solid media. And also, if I could, I would probably use optical density reading for more accurate measurement of growth dynamics of L-coumarins. So finally, I would like to thank the parent support group of Saint Maur International School for partly funding for this work, and also Miyu Shu, who's also here presenting her poster in the other room, for her support and ideas during my study, and to Dr. Suzuki for her technical support, and finally to Dr. Ward for his supervision. And that will be the end of my presentation. Thank you.
Bye.